just going back to the original spirit of the Gabriella Miller grant, there were some data elements expected as part of the X01s to submit, and those were in demographics, clinical information, phenotypic information, including measurements that are related to the primary outcome of the study, and family medical history when possible. Now, since these are retrospective studies, obviously there's going to be some studies do not collect all data that would be interesting to everybody who would like to look at that study. But you know what we're really concerned about in the DRC, in the Gabriella Miller Kids First Data Resource Center, is promoting interoperability of all the cohort data as it comes to us, and integrating and allowing and supporting integrated analysis for people who come to the data resource to use the data. So different cohorts have different levels of phenotypic detail, so that's one challenge we have. Some cohorts may have very basic data, some cohorts may have very deep and detailed data, but we need to make those two cohorts communicate, or at least their data has to be able to communicate on some level. So that's part of the harmonization process. We can apply common classifications to the data. And we've defined a minimal set of phenotype elements for the cohorts, and which I'll discuss shortly. And this information actually helps researchers to connect the cohorts phenotypically across many different levels of information. So to give you an example of how this could work, for those of you who aren't really familiar with directed graph structures, there's every highly detailed diagnosis or disease classification in a well-structured ontology will have information at the tips of the branches of the trees. For example, here you have telecanthus, which and hypertelorism, both of which are abnormalities in the orbital and structure of the skull around the eyes. And if you were not particularly somebody who is really well versed in that kind of structure or that kind of defect, you may not realize right away that those are connected. But the ontology, since we can assign a sample to any one of these characteristics, we can go backwards and allow them to meet up at a higher level where they have commonality. For example, if you have two subjects, 1 and 2, with two different conditions, by using the data structure of the ontology, we can link them back up to abnormalities of the orbital region, coming back to what Melissa was discussing, where you have people with partial or somewhat only partially overlapping phenotypes, but still having similar conditions. So in this way, harmonization to this kind of ontology structure allows people to bring together disparate cohorts with varying phenotypic information. So the main classifications we've decided to focus on in Gabriella Miller Kids First is the NCIP, which is the NCI's thesaurus, which covers diseases, phenotypes, anatomy, and gene function as well. And so we have it at the bottom there covering the entire range. We also have very specific ontologies, one of which is MONDO, which is a monarch disease ontology, and HPO, which was discussed earlier by Melissa, and Ubron for the tissue and anatomy part of for the cohorts that come to us with particular biospecimen or structural defects. For example, someone focusing on particular problems or cancers in certain parts of the body, or defects in certain parts of the body. And then gene ontologies, which are gene function, subcellular location, and biological process. And the gene ontology will cover a lot of the information on the genes that are affected by the variants that we're studying. So oh, my colors are gone. All we came up with a list of minimal clinical phenotypic data elements that we are requesting, if possible, the X01 investigators give us. And if you're a new X01 investigator, this is basically the fields that we would request as a minimum from your study. Family ID, participant ID, everything on the left is pretty much demographics. On the right are the basics of the phenotype text. We don't request that people come up with HPO, NCIT, and Mondo, and which we also don't mention Uberon in this list, but it's one of the ones that we have for specifics of anatomical location. And the reason we don't ask for that is because we don't expect, we encourage it, but we don't expect everyone to feel comfortable with mining HPO and NCIT themselves, and we can certainly provide help for people doing that. So here's an example of a sheet that was filled out by one of our investigators. I know it's a little busy, but I'm going to just zoom in on one particular area here. And this is, can you see? Yeah, you can see that. So this is a group with a group set of multiple defects. So here you have the system ID, a family ID related to that person, their family relationship, in this case is a proband, female, white, she's not Hispanic or Latino, and she has several different birth defects. So these data would then be deposited into Kids First, so that anyone who wanted to study anybody with, for example, congenital diaphragmatic hernia would be able to easily find that person either through a text search or a search on the HPO term. And if their ventricular septal defect, for example, is a particular type of heart defect, so if you're searching for atrial septal defects or just generally heart defects, this patient will be pulled in because of the fact that they are in a child term of heart defects. So beyond the minimal phenotype, there are a lot of studies that come in with different types of data beyond the data that I've seen here. For example, some of you may ask yourself, well, I have a study that has much more data than what you're asking for, and we certainly want that data. We're not saying that the minimal data is what you should just submit. We can certainly take everything. Some studies, for example, is event-driven data, meaning that there be certain encounters with the patient or different events in the patient's medical history that are part of the phenotype and need to be, that are part of the study of the phenotype, and we certainly welcome that data. There's also people who will have additional data that goes beyond the minimal phenotype sheet into much more detailed phenotypes, lab results, etc. And we will certainly work with those groups to create custom solutions and get that data into the repository. So the minimal phenotype sheet suggests at least one ontological term for each subject phenotype. We encourage it. But as I mentioned, we can work with investigators as needed. And if you would like to do this yourself, or you would like to explore how to do this yourself for your data or other related data you'd like to deposit with or work with Kids First, there are public services to help you with that, to seek appropriate terms. So here's an example of a rapper who's got a pectus excavatum. And actually there's an actual web page out there with example of 17 celebrities with this condition, and I think Jeff Goldblum is one, and he's all kind of laid out in his little open shirt, which is but it's interesting. It's some call it a minor birth defect, and there are several other terms associated with it. And here is the Ontology lookup service, which you can Google and find for yourself. The ontologies on the left-hand side, you can see that there are several ontology types that are covered when you do a search. NCIT is among them, as well as there's Mondo and HPO. And when you do that kind of search, you can look down the list and you can see there are several different definitions for different pectus excavatum, and also there's syndromes associated with it. So it does sometimes take an expert in their particular disease. So someone who may not know the difference may choose a term that may not be exactly what the investigator initially wanted or decided on. So we'd like to work with the investigator or have the investigator initially look through their ontological categorizations before we do. So the workflow of how we work, phenotypic information is taken from the source, and then the data is submitted in whatever form, usually the minimal phenotype sheet, and any additional data, and we review the data. And this can be an iterative process. We can actually go through and help ontologize the phenotype and clinical data using the selected classification standards, and we can go back and forth with the investigators on this stage. Finally, under final review, the data is ingested into the data platform, and I'm leaving out a little bit here where we would probably, if there's any additional fields that need to be added and things like that, we could add those, but this would go into the data platform and be later displayed in the portal. So I was going to show you the 2018 data summaries. These are fresh off the data resource. Natasha Singh, who works with Allison, was able to get me some of these as of yesterday. So these are really hot and fresh. So, so Chung, Vitali, Marizita, there's multiple cohorts. For example, Chung's cohorts include all 3 years. So currently, for congenital diaphragmatic hernia, she's up about 2,300, almost 2,400 subject samples with about 755 probands. So the way you read this is, and I'm sorry I don't have a little key at the top, I guess I lost that, but it's the total number of samples. The first number is the probands, the second number are the family members broken down, so you can see how many probands are in each group in the first number. And for example, the neuroblastoma set in Maris's group had originally 639 probands, and the same with Mulligan, 44 probands, but no family members. So there are going to be some proband-only cohorts, and then there are some with extended families sets that will have more family members than you would expect from just a mother and father pair of the proband. And there's a demographic summary. There are some complexities in how we're reporting things like ethnicity or race. Some of it is, and I've collapsed some of this, the unknown and not reported in race. There's also a not collected in ethnicity. And there's also, I also put in there the I'm not able to collect. There are some rules in certain countries that you're not able to collect certain information, demographic information. So I collapsed that into the unknown, not reported fields. But essentially, we have a lot of data, and this data is mostly complete, or it should be all complete when it was ingested. But for race and ethnicity, we have a lot of information as well. So I just wanted to briefly, there may be some people who want to know, how do I actually look at what fields or what information is in the phenotype data? And you can go to the data resource portal, and on the left side, I just highlighted the diagnosis text, but if you scroll up and down, there's a lot of ways to access the clinical data. You can select studies, and you can get information on, you can select whether or not you want proband only, and you can see here there's several diagnosis types that are in common across all the different cohorts. This is all cohorts, by the way. I didn't select any one particular cohort. So if you select any one of these, you'll zoom into certain studies, etc. But for epidemiologists who want to know what the study structure was, or whether or not there were certain types of birth defects collated across different, co-ascertained across different cohorts, or what other types of information is in there, there is sometimes other information besides what's shown here. And to do that, you can actually go into NCBI, or access dbGaP. So I'm just going to show you, for those who have not encountered this yet, there is a NCBI Kids First Umbrella Bio Project repository for all of the Kids First data. And if you zoom, I'm not going to show you details now because I would sit here and click several times, but if you follow the breadcrumbs of essentially going through the umbrella project to your project of desire, and then from there into dbGaP links, you'll end up at a page like this, where in this case I've chosen one of Wendy Chung's cohorts in congenital diaphragmatic hernia. And you'll see there's two links here: download variable report and download data dictionary. And both of those will have details on what's actually what data has been submitted with the cohorts and what data is available. And so, for example, the data dictionary for a data table, when I click on data dictionary, you'll have information here on for example, what kind of birth defects or types of birth defects were selected. There's information on central nervous system defects. Sometimes there's some developmental disorder information. And there's other information on whether or not the child is alive or not. And you can get those from the portal. Sometimes you can get them from here to show you how the study was structured. And then there's information in their variable report as well with counts. So this is sometimes helpful for people to go in and start seeing what kind of data is available specifically for a particular study. And also, I wanted to point out that when you do log into the portal and you go under support, there is an update on the different studies and the type of, and the harmonization state of each study and release notes. So for those of you interested in certain cohorts, you may want to look through the release notes on the release data.